Hey everyone, it's 1 o'clock, so I think we'll get started. Good afternoon, morning, evening, wherever you're watching in from. So happy to have you here. My name is Amanda Moore, and I'm the CEO of the Angelman Syndrome Foundation. And thank you so much for joining us today. We know that sometimes it's hard to get away in the middle of the day, but we think this is really, really important information that you'll be excited to hear about today. So we're excited to welcome MavriX Bio team to share an overview of their Ascend AS program. I'm super honored to be here with my friend and partner, Ryan Fisher, who is the COO of FAST, and I will pass it to him for some housekeeping information.
Great. Thank you so much, Amanda. And hello, everyone. Welcome. So just a little housekeeping before we get started. You'll see the Q&A function on the bottom of your screen where you can submit questions throughout the webinar. We'll try to get to as many live questions as possible. We actually received over 100 submitted questions prior, which is great. We'll be sure to ask a number of the ones that were consistently submitted prior to today's webinar. And of course, we're going to forward all live questions to the MavriX team following the webinar, which will inform future presentations, FAQs, and educational materials. So with that, I'm going to pass it back to my friend Amanda, who is going to introduce our speaker.
Awesome. Thank you so much, Ryan. Well, I'm sure that you don't want to hear Ryan and I speak. You're more excited to hear about what's happening today. So I'm going to go ahead and pass it over to Allison Brandt, who is the Chief Development Officer for AS2 Bio. And she also serves as the Chief Scientific Officer for FAST. And she's also the co-director of ABOM. And most importantly, I think, she's mother to Quincy. So Allison, I will pass it on to you.
Thank you so much, Amanda and Ryan, for the introduction. As you said, and for those of you who I don't know, my name is Allison Brent, and I am the Chief Development Officer at MavriX Bio, which is a portfolio company of an accelerator that was launched by the Foundation for Angelman Syndrome Therapeutics, or FAST. That's called AS2 Bio, as Amanda mentioned. I also serve as the Chief Science Officer at FAST, and I am the co-director of the Angelman Syndrome Biomarker and Outcome Measure Consortium. I am employed by AS2 Bio, and the material in this presentation contains scientific information about an investigational medicine that's not approved by the FDA or any other regulatory body. That's very important. The information provided is exclusively for informational purposes by MavriX and is not intended to provide any medical advice. And questions about medical care really need to be discussed with your clinical care team. So with that, who are we? So MavriX was launched by FAST in 2024 based on promising animal data using a potential gene therapy from a fast track grant that was funded to Dr. Jim Wilson at the University of Pennsylvania. And this program was initiated to develop a possible gene therapy that was subsequently licensed by MavriX Bio from Penn so that we could take the next steps to advance that program with the goal of getting it into human clinical trials. So our team at AS2 Bio and MavriX includes multiple people that have had an unwavering commitment to Angelman syndrome, consisting of incredibly committed individuals, many of whom have a very long track record in industry and who are here really to advance drug development, specifically focusing on Angelman syndrome. We have been able to apply our team's understanding of AS, as well as the learnings from caregivers' lived experience in multiple surveys and meetings that many of you have attended, to support drug development specifically related to Angelman syndrome. And that collaboration in every facet of drug development has been incredibly critical to advance this investigational therapy called MavriX 220, from discovery at Penn through the support of FAST to this first-in-human clinical trial, which we call Ascend AS clinical trial. Within that ecosystem of drug discovery, which involved an academic collaboration with Penn and community engagement through FAST, MavriX sponsored additional preclinical studies to help assess the effects of MavriX 220. And these studies included assessments of response in the Angelman syndrome mouse model, as well as other models of safety. The Angelman syndrome community has been working together in a pre-competitive way to advance endpoints and biomarkers to help design clinical trials for Angelman syndrome. And this program is one of those clinical trials that leverages these learnings from endpoint and biomarker consortium called the ABOM, which you can see here. In addition, we have leveraged all of the incredible work by the Angelman syndrome community, and that's you, including all of your efforts in the Angelman syndrome natural history study that has been ongoing for decades in different stages since 2006, starting with support from the NIH, followed by support from the FDA, and more recently by support of the collaborative effort of the ABOM. In addition to these collaborative efforts, FAST launched this accelerator, AS2 Bio, with the goal to help advance different therapeutic platforms for Angelman syndrome, some of which are at very early stages in preclinical research. But the one we're going to talk about today is an investigational AAV gene replacement therapy. And MavriX Bio is a portfolio company of AS2 Bio that is sponsoring this Ascend AS clinical trial for MVX 220. So I'm sure you're wondering, if you don't already know, what is gene therapy and what is MVX 220? So let's walk through this a little bit. So let's start with just explaining what is gene therapy. We all hear that word a lot. Adeno-associated viruses, or AAVs, are small viruses that are generally considered not to cause disease in people. They've been modified and used in hundreds of clinical trials to carry different genes to cells where the gene might be missing or might not be functional. And AAV is one of the most common carriers or vectors used in gene therapy. In general terms, here are the steps on how this is done. So you can see here, the first step is that a missing or non-functional gene, in Angelman syndrome, that gene would be UBE3A, is packaged into a carrier. And in our case here, that would be the AAV vector. The second step is that that vector carrying this gene is then delivered to a patient. And this, as you could see here, can be done in various different ways. One way that it could be delivered is intravenously or IV in a vein. And that travels throughout the body. Another way that it could be delivered is right into the cerebral spinal fluid, or the CSF, which is that fluid that surrounds the brain, in order to target the cells of the brain, like neurons. Then once that virus carrying the gene reaches those target cells, like in AS, it would be targeting those neurons of the brain, the vector then delivers that therapeutic gene into the cell, where it can create the protein that is missing or non-functional. And in the case of Angelman syndrome, that is UBE3A. So I'm sure you're wondering if this has ever been done before. Well, actually, over 2,500 clinical studies using various types of gene therapies have actually been tested in humans since the 1990s. And as of July of 2025, so just a few months ago, when a big publication came out showing that rare disease is the most common use of gene therapy, with more than 50 AAV gene therapies that are currently in clinical development for various different types of genetic disorders. And as you can see here, over 500 active clinical programs are in development for rare disease using gene therapy, and that's followed by cancer and then other types of systemic disorders. So what does it mean for Angelman syndrome? And as I'm sure most of you know, Angelman syndrome results in the loss of expression of a single gene called UBE3A. And if you look at this graphic here, what you can see is that there are two copies of every gene. There's one copy here, the maternal copy that comes from mom, and a second copy here, the paternal copy that comes from dad. Interestingly, the father's copy of this gene is naturally silenced only in neurons or brain cells. And due to that silencing mechanism, all of us, including you and me, only have expression of the mother's copy right here. And that's only in the brain. In the systemic periphery, we have mother and father being expressed. But those living with Angelman syndrome, that maternal copy is either missing or not functional. And when that happens, you have no expression of the maternal UBE3A gene and silencing of the paternal UBE3A gene. Therefore, there ends up being no expression of UBE3A in neurons of the brain. All genotypes of Angelman syndrome result in this maternal UBE3A deficiency or lack of function in the brain. Whether that individual has a deletion, a mutation, uniparental disomy or UPD, an imprinting center defect or ICD, and even mosaicism. It all results in a deficiency in UBE3 expression in neurons of the brain. And since individuals with AS have a lack of this expression or function, a gene replacement therapy is intended to replace this missing or non-functional copy of the maternal UBE3 gene in neurons. So what exactly is MVX 220? Well, MVX 220, it's an investigational gene replacement therapy. And it uses that AAV virus we just discussed as a carrier to deliver a copy of this human UBE3A gene, which is packaged inside that virus, as you can see here, to the neurons of the brain in an effort to replace that missing or non-functional copy. It is intended to provide a way for those neurons of the brain to make a small functional UBE3A protein. This AAV is delivered into the spinal fluid by a small needle, as you can see here, at the base of the skull, and it is inserted and injected into that fluid. The hope is that once it goes into that spinal fluid, which is bathing the brain, the AAV will get into the brain cells and produce that missing copy of the UBE3A gene. So the history of this program started with a rigorous screening and development program in a collaboration between FAST and Penn under the leadership of Dr. Jim Wilson, and that began in 2017. That led to the discovery of MVX 220, which was tested in the Angelman syndrome mouse model, where UBE3 protein was present in neurons of the brain after being administered, resulting in an improvement in several of those symptoms in the AS mouse. MVX 220 was then tested in non-human primates or monkeys, and these animals did not have Angelman syndrome. They were healthy animals, but testing was done in this large animal brain in an effort to evaluate the safety and the distribution of MVX 220 to the neurons of the brain. And together, these animal studies were submitted to the FDA along with manufacturing information and a clinical trial plan as part of an investigational new drug application, which is called an IND. And all of this information was submitted for review, allowing us to initiate the FAST clinical study, which is now excitingly open for enrollment. So let's talk a little bit about the details of the trial, which is, I'm sure, what you've all been waiting for. So MVX AAV 101, also known as FAST AS, is a multi-center, open-label, phase 1/2 study looking at the safety and efficacy of MVX 220 administered by that intracisterna magnet injection to participants living with Angelman syndrome. This is a first-in-human study that is primarily focusing on assessing the safety of MVX 220, both in adults and children living with Angelman syndrome. It is designed to understand the safety, the tolerability, and the potential efficacy of this type of gene therapy. The clinical outcomes selected were based on the learnings from the collaborative efforts of the ABOM, with additional community input from surveys and the externally led patient-focused drug development meeting that occurred in April of 2025. And these efforts allowed us to design this clinical trial to assess outcome measures that deeply matter to patients and their families. Open label means that there's no control arm, so everyone enrolled in this study will get the treatment of MVX 220. And this study uses a single dose level, meaning that everyone will get the same dose level, and that dose is based on each individual's brain size that will be determined by a pre-dosing MRI. So as mentioned, MVX 220 will be delivered into the cerebral spinal fluid, or the CSF, into a small space here sitting at the base of the skull. It's injected into this area, which is called the cisterna magna, using a very small needle. And this procedure is called an intracisterna magna injection, or you might hear it as ICM delivery. This procedure is performed under general anesthesia with image guidance using a CAT scan. And all doctors that are performing this procedure are specifically trained for this study. So let's cover the study design. So the study is divided into 3 different cohorts. The first cohort is the adult cohort, enrolling participants ranging in age from 18 to 50 years old. Each adult participant will be dosed sequentially, allowing for a required follow-up period between each patient in order to ensure we're evaluating safety. These required follow-up periods result in one patient being dosed at a time, with all of the available safety information reviewed by a safety committee prior to dosing the next patient. After all participants in a cohort are enrolled and dosed, then there's an additional safety review by an independent safety committee to determine if we can enroll the second cohort right here. Cohort 2 will enroll children 4 to 8 years old. Patients again will be dosed sequentially with safety follow-up between patients, as no patient will be dosed on the same day. Once all patients are enrolled from cohort 1 and 2, additional data review will be conducted by that independent safety committee. And after that review, to ensure all acceptable safety profiles have been met, an optional cohort called cohort 3 might be enrolled. And participants in cohort 3 can range in age from 4 all the way to 50, allowing for enrollment of both pediatric and adult participants. And MavriX will decide if cohort 3 will be enrolled at a later date based on that data review, as mentioned. So how are we going to determine the dose of this gene therapy? The target for Angelman syndrome is to deliver MVX 220 to the brain cells or the neurons of the brain. Because MVX 220 is dosed directly into the brain fluid by ICM injection, the total dose given is based on the weight of each individual's brain, not the weight of their body. This is important. We use an MRI at screening to estimate that individual's brain weight, and that allows for a total dose to be calculated for each person based on their specific brain size. Most IV or intravenous gene therapies don't target the brain, but they actually target other parts of the body, which is much larger than the brain. In these cases, the total dose determined is based on how much the patient actually weighs. So what you can see here is that the average brain weight of a child between 4 and 8 years old may range between 2.5 to 3 pounds, where the average body weight of a similar child, 4 to 8 years old, may be 33 to 57 pounds. So you can imagine that the amount of gene therapy vector that is given when based on total brain weight is generally lower than when it would be based on total body weight. Therefore, generally, by dosing directly to the brain based on brain weight, it requires a lower total dose than what one might give if they were giving this intravenously as a gene therapy. So what are the objectives of this study? As mentioned before, the primary objective is to evaluate the safety and tolerability of a single dose of MavriX 220 in individuals living with AS. The secondary objective being to measure levels of MavriX 220 in the blood, in the spinal fluid, and to evaluate the potential efficacy of MavriX 220 based on various clinical outcome measures. So what are they? These outcome measures, these key outcome measures, are very important, and they include assessments that measure the main symptoms of AS that you, caregivers and clinicians, have determined to be among the most important, including communication, gross and fine motor skills, activities of daily living, quality of life, sleep, challenging behaviors, cognition and learning, and we'll also be evaluating various biomarkers. So assessment for potential target like EEG, MRI, and potentially measuring different proteins in body fluids like blood and CSF. So let's cover some of the important aspects of enrollment. The study is designed, as mentioned, to include patients that are 18 to 50 years old in the first cohort and 4 to 8 years old in the second cohort. The genotypes that are permitted include large deletion, as well as those with UPD, uniparental disomy, and ICD, imprinting center defects. Each individual must be able to ambulate independently, which means they don't rely on a device such as a gait trainer to walk. And if participants take medication to control their seizures, they need to be on a stable dose of that medication without any changes for a short period of time prior to enrollment. If they use a diet to help with epilepsy, they must be on a stable diet as well prior to enrollment, and that would be a diet like a ketogenic diet or a low glycemic index diet. Individuals will be excluded if they have other clinically important medical challenges or problems that might not make them a great candidate for the study. For example, if an individual with AS has something like diabetes or another genetic condition, they would not be able to enroll in this study. If they have abnormal blood values that could interfere with assessing the safety of MVX 220, or they have known serious infections, they would not be able to enroll in this study. If they have any known history of an autoimmune disorder or significant inflammatory conditions, or if they've been previously treated with an antisense oligonucleotide within 1 year of enrolling in this study. So what that means is that it is okay to have been exposed to an antisense oligo or an ASO for Angelman syndrome in another study, as long as it was with more than a year that the ASO was stopped. And if the study doctor were unable to get that MRI and estimate the brain weight and to check the cisterna magna area for injection of that participant in order to make the decision that they are safe to have that injection, they would not be able to be enrolled in this study. If they're unable to receive steroids as part of the study, they would not be able to enroll. And we'll certainly cover a little bit more on the steroids in subsequent slides. These are not all the criteria, please keep in mind, just some of the key items. And so if you're considering enrolling, I would encourage you to contact the study investigator and they'll go over and review the full list of enrollment criteria with you to help you make a decision on eligibility for your child. So the study does involve a number of visits to the hospital that are more frequent at the start and become much less frequent to try to reduce the burden on families and participants over time. The study will be 5 years in duration, and this is mainly because the regulatory agencies like the FDA require us to conduct long-term follow-up to help understand if gene therapies are safe and provide long-term effects that help the individual. So to highlight some of these visits, what you can see here is that the screening period requires an MRI to determine eligibility, as we mentioned, and a spinal tap or a CSF tap to collect fluid for laboratory assessment prior to dosing. The MRI and spinal tap can happen at the same visit, so the individual does not need to go under anesthesia twice for those activities. Each individual will have an EEG performed and various clinical outcome assessments, either at home by an interview, recorded on a tablet at home, or performed in the hospital prior to dosing. Various laboratory assessments will be done in the blood as well prior to dosing. And once the participant is enrolled, they will be dosed with MVX 220. And after that, they will need to remain in the hospital for 3 days prior to dosing for careful monitoring to ensure that they're tolerating MVX 220 well and that they're not having any immune reaction or adverse reaction to this gene therapy. Once an individual is discharged from the hospital, they will still need to be seen daily for an additional 3 or 4 days for safety laboratory assessment and for monitoring by the study doctor. After that, over the first year, there will be intermittent visits to the hospital to see the doctor mixed with telehealth visits from home. There will be various laboratory tests, mostly blood draws, that will be performed. These, to help really lessen the burden of needing to visit the hospital, many of these assessments and laboratory tests can be done at home from local labs near the patient's home or by telehealth. Over the second year, the frequency of visits to the study site will go down even further and will include doctor visits only twice a year, some telehealth visits intermittently with the doctor, as well as some local laboratory testing that can be done near home. And then from years 3 to 5, the patient will continue to be followed intermittently with a yearly doctor visit, telehealth visit, and then 1 local lab visit and 2 scheduled calls from the site to check on safety. So the study is long, but the need to visit is reduced dramatically over time. So what are the potential risks and what is the monitoring plan for this study? It is really important to recognize that all investigational therapies carry both short and long-term risks that families must understand before participating in any clinical trial. For gene therapy, Different viruses, meaning different viral vectors, and different methods of delivery of those viruses will carry different risks. So we're going to highlight those related to this type of AAV virus that is used with MVX 220 and this type of delivery, the ICM delivery route. So there are some procedure-related risks with ICM or intracisterna magnet injections. These risks, albeit rare, can include things like headache, vomiting, bleeding, and infection. And in order to minimize those risks, there will be careful monitoring with a trained anesthesiologist present during the procedure. And this procedure will only be performed by a doctor, like a neurologist or a neurosurgeon, who is specifically trained in this ICM delivery method by an expert who has performed the procedure on other gene therapy studies and trained many other doctors around the world. A pre-procedure MRI is being done, as mentioned, in order to check that each participant is a good candidate anatomically to undergo this injection. There's also drug-related risks directly related to the MVX 220. An immune reaction or an overactive immune system can happen with any gene therapy, just like any other virus. But because you are injecting these viral particles into the body, even though this vector has been changed to take out, the bad parts that could theoretically make a patient sick, we have to still consider this as a potential risk. And each individual, for that reason, will be required to take oral steroids. And that will be for a few days prior to the dosing and for approximately 3 months after the dosing in order to minimize the risk of an immune reaction to the gene therapy. Now, it's important to recognize that steroids can lower the immune system capacity to fight infections, and this is called immunosuppression, which can result in a temporary higher risk of infections. It's very important that individuals are able to take steroids to keep taking them throughout the course of the study until the study investigator or doctor tells them to stop. Steroids themselves can have their own risks, including things like mood changes, sleep changes, as well as appetite changes. This is also why we're excluding participants who have a known history of any serious immune conditions and why steroids are used to lessen the possibility of an immune reaction. There will be careful and ongoing monitoring that will occur throughout the course of the study, like assessing blood work, in order to ensure that the body systems like the kidney and the liver are all functioning well. This type of monitoring and steroid use is standard for all AAV gene therapies as well. Monitoring for things like liver toxicity is incredibly important because the liver is where the AAV virus likes to go. The steroids also help to minimize any potential liver toxicity. Sensory nerve effects have been reported in animal studies with AAV gene therapy. So CNS or central nervous system delivered AAV gene therapies generally monitor for this risk. We'll be monitoring throughout the study, assessing gait, sensation, balance, and movement. And finally, understanding the uncertainty of the durability of an AAV gene therapy, knowing that individuals that receive an AAV gene therapy today may not be able to receive another type of AAV gene therapy in the future. The participants in this trial will be followed for at least 5 years to best understand the long-term safety and that potential long-term efficacy. So what about cost and travel reimbursement for this type of clinical study? A service will be provided to support all of your travel needs and reimbursement for all families that are enrolled in this clinical trial, including a concierge service that will help schedule hotels, flights, transportation, reimbursement for things like food and parking, mileage, gas, tolls, all of that will be provided. And we will ensure there is minimal to no out-of-pocket expenses for individuals and their families enrolled in this study by using a reloadable payment card to support your financial needs. So with that, I would like to thank everyone that has been involved in the process of this study and the development of MVX 220. particularly the team at the University of Pennsylvania, now MavriX Bio, the Foundation for Angelman Syndrome Therapeutics, the Angelman Syndrome Foundation, the Angelman Syndrome Natural History Study, the entire team at MavriX and AS2 Bio, but most importantly to you, our community. We are so very excited to share this opportunity with all of you. So with that, I'm happy to answer any questions, and I will turn it back over to Amanda and Ryan.
Wonderful. Thank you so much. It was really, really helpful. Covered a lot of questions that had come in prior to the webinar. I want to start with the inclusion and exclusion criteria for the trial. Can you explain a bit about the rationale for the genotypes chosen, and can you specifically comment on the exclusion of mutation?
Of course, of course. And that's a really great question, Ryan, and I'm happy that it's been asked. So as we mentioned, MVX 220 is an investigational gene therapy, right? It's designed to deliver the human UBE3 gene to neurons of the brain by a single injection into that area of the skull, at the base of the skull called the cisterna magna. And it is delivering these viral particles to carry a healthy copy of that gene. Once it is injected into the fluid, the virus and the gene reach the neurons of the brain with the goal of replacing the missing copy of the maternal UBE3 gene that is either not functional or not present. Individuals who have a deletion or UPD or ICD genotypes have a genetic variation that in almost all cases results in no UBE3 protein that is being made in neurons of the brain. Individuals who have a mutation genotype or a point mutation can have different types of these gene variations that in most cases also result in no UB3 protein that is being made in the brain. But in some cases, a modified UBE3 protein can be made that does not work well as it should, but it's present and it does have some function. And individuals with a mutation genotype who are making this type of modified protein may not benefit from additional healthy protein being delivered to the brain. So even though the situation is likely quite rare, it's hard to predict which gene variants make this type of modified UBE3 protein. And therefore, because this study is so small, we had to make the decision to exclude these individuals at this time, just until we can better understand more about which mutation subtypes might be good candidates for gene replacement therapy and which might not.
Okay, so maybe a matter of timing. Thank you, Amanda.
Yeah, thank you, Allison. I know you kind of covered this, but the questions have come up again, so maybe we can answer it one more time. But why only 12 patients for the study?
That's a great question. We would love to include a lot more. So MVX AV101, as we said, is a first-in-human study. It's a phase 1/2 study with a primary objective of really understanding the safety of the treatment with MVX 220. It is very common for phase 1 studies that are first in human to include a real small number of participants because the goal really is to learn about the safety of the drug and ensure that only a limited number of participants are exposed to that drug while the safety data is collected and understood. In gene therapy studies, the administration procedure can require hospitalization and significantly close follow-up and long-term monitoring. Sometimes it's 5 years, sometimes it's 15 years, depending on the type of gene therapy. So the study size is often very, very small compared to other types of drugs to help make it easier for the sites to follow these individuals long-term.
Thank you, appreciate it. Ryan?
Great. So when you look at how the study rolls out, why are you starting in adults?
That's also a great question, and I'm very happy to answer that question. So the first cohort, as mentioned, will include adults, and really it's to help understand the safety of treatment before enrolling younger children in the study. And this is not uncommon in first-in-human studies when a disorder affects both adults and children. But we also feel really strongly that we need to understand how older patients may respond to treatment with a gene therapy and if they derive benefit, as we strongly believe there is a prospect for benefit for both adults and children. And emerging data from the investigational studies of the antisense oligonucleotide drugs for Angelman syndrome specifically really do suggest that adult patients are having a positive response to treatment. So we felt that that was, really a strong consideration to include adults in this clinical trial. And certainly after review of the data of cohorts 1 and 2, then MavriX may enroll an optional third cohort and include even more adults in the study, as long as we can show that it's safe and we're providing benefit.
Excellent.
So as you probably know, Allison, a lot of the questions that are coming across in the chat and that have come to us before is the question of where these studies will be conducted.
Of course, of course. And that is something that we all strive to be able to get that information. So we're still in the process of activating sites for this study. But just, for what we know at this time is that the plan is to include sites in the United States and Canada at the start. Rush University Medical Center in Chicago is now recruiting participants. They are open for enrollment. And we plan to add additional study sites on the East Coast, the West Coast, another in central US location, as well as in Canada. And the details on the study design and all of these will are currently available on clinicaltrials.gov. But we'll also update the listing on clinicaltrials.gov of who's active as they become available. And so keep your eye on that.
Thank you.
Excellent. So obviously we're a tight global community. And there have been a number of questions around, will families be allowed to travel internationally to sites to participate in the study?
Great question. So the protocol for this study does not require that participants live in the United States or Canada. It does not. However, there are requirements for frequent lab testing over the course of the study that are intended to be performed at a local site that's close to the participant's home to reduce study burden, and the need to visit the primary site during the study is required. And so MavriX will not be able to add lab test sites outside of the US or Canada, which means that frequent trips to the primary site would be required, and international travel may be very difficult for families over a 5-year study period. So if you are considering enrolling your child in this study, you should definitely talk about those study logistics with the trial team at one of the trial sites and make the best decision for your family.
Great, thank you.
So a couple questions. I know you addressed this as well, but I think it's always really good to remind us the length and the time that these, trials can happen, or the time in between. So how long until cohort 2 can actually start?
Never soon enough, right? That's what we always feel. I feel it too. So cohort 2 will start after all participants in cohort 1 have received treatment. And not only received treatment, but have been followed for several weeks for safety monitoring. It is so incredibly important. Safety data from the first cohort of adult patients must be reviewed by an independent committee before cohort 2 can be started. So it cannot be rushed. These things just take time. The follow-up period and safety committee review are absolutely required by the protocol to help ensure that safety of participants is our #1 priority in this study. So we hope to be able to start cohort 2 in the second half of 2026, but really it's going to depend on enrollment and recruitment in order to be able to give you a better sense of timing.
Thank you.
Okay, so this is an important question given all the activity in the Angelman syndrome space. In terms of how would participating in the trial affect a person's ability to participate in a future clinical trial?
It's a great question too. And these are, I'm so happy the community is asking such wonderful questions, because these are the questions you should be asking. So because gene therapy is meant to make a permanent change in the body, and in this case, it's delivering it is using a viral vector to deliver that copy to neurons of the brain that's intended to make that UBE3A protein express. Most other clinical trials will exclude individuals who have been treated with a gene therapy because it won't go away. We cannot speak to what the eligibility criteria will be for all future studies in AS. That's going to be up to those sponsors. So you should discuss this with your child's doctor or the study investigator if you're considering enrolling in a study of a gene therapy for AS or any future study.
So you talked a little bit about that vector just now. So why was that AAV vector chosen? And can you tell us a little bit about the data on this vector with other gene therapies?
Of course, of course. So this specific type of virus, the viral vector that will be used in this study, is called AAV HU68 or HU68 capsid. And this was developed by Dr. Jim Wilson at the University of Pennsylvania. It was chosen based on their studies in animals, which shows that this vector is able to get into the neurons of the brain. And it has been used in clinical trials of children, even very young children, with disorders like early infantile Krabbe disease, GM1 gangliosidosis. Each program uses this AAV-Hu68 viral vector with the gene cargo inside of it that's specific to that gene of that disease, so not UBE3A. So in both programs, the AAV-Hu68 viral vector was administered exactly as we're doing by a single ICM injection using the same viral vector.
Thank you.
That's really helpful. We did see this question a few times. With gene therapy, is there a risk for overexpression?
That's a great question as well. So MVX-220, obviously, as we mentioned, is an investigational gene therapy designed to deliver the human UBE3 gene to neurons of the brain using this AAV delivery vehicle. And so once it's injected into the fluid that surrounds the brain, the virus and the gene should reach those neurons with the goal of replacing the missing or non-functional copy of the gene. Individuals with deletion, UPD and ICD, that have these gene variations that are producing no UBE3A protein, we would like to deliver the UBE3A gene and ultimately the protein to those cells of the brain. Since a viral vector will not reach every neuron in the brain, to every part of the brain, the risk of overexpression of UBE3A, meaning making more UBE3A than a neurotypical individual might already have in their brain, is very, very low. In animal studies in the AS mouse, for example, we did not see any evidence of overexpression of UBE3A using specifically MVX-220 at the dose level proposed for this study. So we don't believe that that's going to be a major concern.
So I know you touched on this earlier, but I think it's always good just to kind of level set and understand how gene therapy is happening across the board. So can you discuss a little bit on how gene therapy has been used in other rare disease areas?
Sure, sure. Gene therapy has been used in patients with many other diseases, like things like spinal muscular atrophy or SMA, Duchenne muscular dystrophy, hemophilia, many, many rare genetic disorders. And in the US, there are over 20 approved gene therapies for treatment in humans, and over 200 clinical trials have studied the AAV specifically like this type of viral vector. And like I said in the slides, a recent report from a group called the ASGCT, the American Society of Cell and Gene Therapy, that collects information on gene therapy development programs around the world, just showed that currently there are over 500 clinical trials of gene therapies ongoing for humans for rare diseases. And so this is certainly not the first of its kind, and this has been happening for decades.
Thank you.
Great. Another common question we've gotten is if your loved one is or was on an ASO, and you talked about this earlier in your slides, but can you talk a little bit more about it, and can they enroll in the trial if they are or were on an ASO?
Yes, great question. So if your child right now is actively participating in another clinical trial with an investigational ASO, and they're actively being dosed, they cannot enroll in this clinical trial for MVX-220, not at this time. If your child was previously enrolled in a clinical trial and received an ASO, but no longer receives that treatment with an ASO, they might be eligible for the study, provided that it has been stopped for at least a year since they were last treated. Detailed eligibility criteria is posted on clinicaltrials.gov, and I encourage you to talk to the study doctor so that they can ensure, if you're interested and you have stopped that ASO, that enough time has passed and that they would be a candidate to enroll in this clinical trial.
Yeah, those discussions with your physician are critical here
Yeah.
as we navigate.
So Allison, we have a lot of questions coming in the chat, and I just want to remind everyone that we're going to do our best to get these answered. It probably will be afterwards once we can gather all of what we have as we're running out of time. But we have a few questions left that we will try to do our best to get answered here. And one of them is, if you can explain a little bit more, so why no children less than 4, and when do you plan on maybe going to children less than 4? Does that make sense what I'm asking?
Yeah.
Okay.
So the reason we chose children 4 to 8 years old is because what we know thanks to the Angelman syndrome natural history study, is that individuals with Angelman syndrome, we can all be really grateful that they never lose skills. They gain skills over time, generally, particularly from age 0 to 4. Then they become flat. They don't lose skills. They do not regress. So firstly, 4 is a great age in which it's as young as possible where there is a flattening of their skill acquisition. So if we improve upon that skill acquisition, we can get above their standard baseline where we have a lot of information on those kids that we would be able to show an improvement to natural history. In addition, it's really important that individuals are able to walk. And so the reason for that is a fewfold, but they can't use, ideally not a gait trainer. They're not using a handhold. They're independently walking. And 4 is about the age where a lot of individuals, whether they're a deletion or a non-deletion genotype, will gain the ability to walk. And that's very, very important because we're using gait as an endpoint, looking at improvement in gait. And it's important that we look at that from a safety perspective, because as I mentioned, with the sensory nerve toxicity potential, we want to make sure that we can watch them walk and that their gait is not being negatively impacted.
So
Awesome. You were able to answer 2 questions at once, so good job.
Oh, good.
Ryan,
just
go
to
ahead.
clarify that means if they have a walker, if they use a walker, they can't necessarily participate, correct?
Correct. They have to be independent.
Yep. I know that there was a question about the control group, and if you don't have a control group, how will you assess whether there's a response to the treatment?
That's a great question. Really is. Generally, in a phase 1/2, you don't have a control group. Just like with the ASOs, there was not a control group. There was an external control in which the participants were compared to the historical natural history study that, we have a natural history study back from 2006 all the way till now. And the patients were aligned with the natural history patients to look at their change over time and compare gains, losses, or stability over time with that external control. So that is very, very common for a phase 1 clinical trial, and that's exactly what we're doing here.
So, so another question that just came through is, well, one, can you just explain again, maybe, I know we just talked about the under 4, but can you explain again about the ability to add all ages as part of that optional cohort 3?
That's a great question. And I think the data we get from cohorts 1 and 2 will help us determine what cohort 3 ultimately could look like. So that is an optional cohort. And depending on the data, we will look at everything at hand and make decisions on what ages, what genotypes, and where we want to focus that cohort. And then again, getting that information will potentially allow us to move to the phase 3, which is what we ultimately want, which can be more, we could think about that potentially being more broad based on the information we have. So everything is always on the table, and everything will be determined based on the safety and efficacy information that we see in those first 9 patients that we dose to determine if we need those additional patients or what that expansion might look like.
Thank you.
Great. There was a lot of questions about why you have to use steroids in this study. Can you talk a bit about the use of steroids in this study in particular?
Of course. So for all AAV gene therapies, and most gene therapies in general, you do need to immunosuppress the participants. Now, part of that is, yes, it's a healthy virus, meaning they took the bad parts out of the virus, but it's still a virus, and your body can react to that virus. When it's delivered into the CSF, the body is not getting as much systemic exposure for that immune reaction to happen. Generally, immune reactions happen systemically, not so much in the CSF. But even though it's being delivered into the CSF, it is going to be eliminated from the CSF and get some peripheral or blood exposure. And so in order to prevent any type of immune reaction, meaning the body sees a load of virus and tries to fight against that load of virus, steroids prevent that from happening. And so in all of the AAV gene therapy trials, there's all different regimens on how you can immunosuppress. Generally, most of them include steroids, and some include things stronger than steroids. We are only including steroids based on our route of delivery and our dose level that we're using. And that just needs to be pretty strong doses for the first month. And then those will be tapered down based on the participant's reaction. And that will be monitored very carefully with the primary investigator with laboratory assessments in the blood. So that will allow us to say, everything looks good, we can drop the dose, we can drop the dose, we can drop the dose. And we expect that tapering period to probably last about 2 months after the higher load at that first month.
Great, thank you.
So I know you've mentioned this, but we see the question still coming through, so I wanted to maybe go over it again. Like, so do you have to get multiple doses of this drug, or do you have to get an injection like every 5 years?
That's a great question. So like ASOs, which are synthetic and they only last a few months, and ultimately they get broken down and you have to give it again. A gene therapy, the virus puts that gene into the target cells, hopefully, and that gene will then, in longevity for a long time, produce that protein. And so essentially it's a factory. And so that would not need to be re-injected. Now, only time will tell. We've obviously never done this in Angelman syndrome. So we don't know how long in Angelman syndrome, specifically in a human, this will last. But it is meant to be a single treatment therapy that lasts long term. And that's why this study is for 5 years plus for us to follow out these patients. And that's critical for us to really be able to show how it works and how it lasts. But only a single treatment because it should last. It's not going to wear down like an ASO in that short time period.
Sure.
Great. I know we talked about this at the top of the Q&A, but understandably, we have additional folks asking the question, but can you just explain again why mutation genotype cannot be in this study in particular?
So as mentioned earlier, there's of all of the genotypes that we have, remember this is a very small study, right? It's only 9 to 12 patients. Of all of the genotypes that we have, it's very important when you deliver UBE3A that you deliver it to patients that are null, meaning that they don't have any UBE3A, or there's no risk of competition of a misfolded UBE3A protein to a healthy copy of a UBE3A protein. We know deletion have 0 UBE3A protein in neurons. We know that UPD and ICD, because they're silenced on both alleles, have limited to no UBE3A protein that's being expressed. What we don't know about mutations is which of the mutations are null, meaning they're not expressing any protein, versus those that are expressing protein, but the protein is just not as functional. If you have a non-functional or partially functional protein, ultimately that can result in almost a competition with a newly delivered healthy protein. And that may not allow for us in such a small study to see benefit. And so if that's the case, being this is such a small study, we couldn't risk diluting out the potential effect to eventually get to many, many more patients, which is our goal. So we wanted to ensure we start with getting that initial data in those that we are confident are null. It doesn't mean we won't get to mutation patients. Our goal is to get there. And the community is working hard to understand mutations and understand prediction of who may or may not be producing a protein. But we're just not there yet. And because of that, we felt it was prudent to put safety first and ensure that in such a small study, we started with those that are null.
Okay, great. I think we have time for one more. So are there other AAV gene therapies that are approved by FDA that are dosed via ICM that you can comment on or give everyone a little bit of color on?
Yeah, so that's what I mentioned earlier. There are 2 other programs with this particular Q68 that's been dosed ICM. There are also others with a very similar vector called AAV9 that are also dosed by ICM delivery. So this is not new. ICM is not new. But the 2 programs that are currently that have been in the clinic, and there's another one coming into the clinic right on the heels of this one for a different disorder, are all being dosed ICM. So there have been the person who has trained the most individuals with this procedure has dosed over 100 patients by ICM delivery. And so it's definitely something that there is a track record for. And it started with Jim Wilson's team at UPenn and the procedural doctor who developed it there and who has gone out then and helped to support the training of many others around the world.
We're not first out the gate.
We are not the first out of the gate, that's for sure.
Great. Wonderful. Amanda?
Yeah, I think we are at our time, and we will do our best to grab whatever questions were not answered, and get them to the amazing team at AS2 Bio for them to give their feedback. And we will also work to get this recording together. And we are very, very excited and thankful for this team for all that they're working on and doing for the community. There's a really exciting time and a hopeful time for our community and for everything that's happening. So thank you, Allison, for spending time with us today and answering so many questions to help us understand what this looks like. And we look forward to many more updates as we come down the pipeline. So Ryan, I'll move it over to you to close us out.
Yeah, thank you so much, Amanda. Thank you, Allison. Really was a great presentation. Just a reminder that the FAST Summit's coming up. You can register virtually or join us in person. We'll be asking questions to all companies in our space to make sure we get you the answers you need in order to make informed decisions. Thank you so much for joining us today. As Amanda said, we'll work to get the recording up for folks, and we hope you have a good rest of the day or evening, depending on where you are.
Thank you.
Thank you, guys.